है ना। तो चलो, सो अगेन हम लोग उसके आगे का भी स्टार्ट करते हैं, ठीक है। सो इन द लास्ट लेक्चर मैंने आपको ये बताया था कि काइलो माइक्रोन का फॉर्मेशन कैसा होता है। तो फिर नेक्स्ट क्या है, अच्छा हाँ, इसमें आपको मैंने लास्ट लेक्चर में ये मैंने बताया था कि स्टमक के अंदर जो होता है वो डाइजेशन कैसे होता है। तो मैं जस्ट अगेन आपको मैं जस्ट ये रिवाइज करवा रहा हूँ बिकॉज इसमें से कुछ पॉइंट पर आपको फोकस करना है। ओके, सो रेडी, स्टार्ट करें।
Yes, sir.
Okay.
Yes, sir.
So first is what I think, so we all know na ki what is chyme. Chyme is what? It is a food mixed with the gastric juice and all. ये जो important है, ये आपको दिख रहा है, formation of the proteoses and peptones. Okay, ये important है because these are these are the product which is formed after the breakdown of proteins by the pepsin. ठीक है, तो तीन कॉन्सेप्ट आते हैं, एक आता है प्रोटियोसिस, ओके, द फर्स्ट इज प्रोटियोसिस, सेकंड इज पेप्टोन्स, ये आपको याद रखना है, प्रोटियोसिस, पेप्टोन्स, एंड देयर इज वन मोर टर्म दैट इज कॉल्ड एज पेप्टाइड। तो ये तीनों चीजें स्लाइटली, दीज आर स्लाइटली डिफरेंट टर्म्स व्हिच वी यूज, ओके, तो ये इंपॉर्टेंट है। So what is peptide? Simply these are the short chains of amino acid. For example, if I draw protein, suppose this is a large proteins, ठीक है, ये क्या है, this is a protein. So if this protein is degraded by the pepsin, कौन इसको break down करेगा? Pepsin. Okay, so it will not directly form the amino acid, ये आपको याद रखना है, it will not directly form the amino acids but rather it will form the small intermediate compounds like the peptides. So the small peptides, something like this, जैसे मान लो कि ये जो these small circles are representing what? Amino acids, और ये जो बीच के जो bonds है वो क्या है? These are the peptide bonds, है ना। So ये that they will break down the amino acids in the small peptides. Is it clear? ये इतना clear है?
Yes, sir.
ओके, अब पेप्टाइड का अलग अलग क्लास होता है, जैसे पेप्टोन क्या है, इट इज जस्ट वन ऑफ द क्लास ऑफ द पेप्टाइड। Okay, so in the exam or during the exam they might ask the difference between peptides and peptones. So peptide is, what is peptide? It is a short chain of amino acid, है ना। So this is what? This is peptide. अब peptides के भी अलग अलग क्लास होते हैं, okay, depending upon its nature, its length and all. Okay, so एक क्लास कौन सा है? Peptones. आप समझ में आया? So peptide can be divided into many classes. Out of those class, one class is what? Peptone. ये क्लियर हुआ आपको? ये समझ में आ रहा है?
Yes, sir.
ओके, उसके बाद नेक्स्ट क्या है यहाँ पे? प्रोटियोसिस. सो अगेन दीज आर प्रोड्यूस्ड ड्यूरिंग द हाइड्रोलिसिस ऑफ द प्रोटीन. सो अगेन फॉर एग्जांपल ये जो पेप्सिन है, इट विल प्रोड्यूस द पेप्टाइड्स बाय द हाइड्रोलिसिस. तो प्रोटियोसिस भी क्या होता है? इट इज आल्सो टाइप ऑफ पेप्टाइड्स, ठीक है, बट it is produced just before producing the amino acid. So it has these are very small peptides, like इनका size जो होता है, these are very small, something like this. So if this is broken down, it will produce the individual amino acids. Okay, तो अगर मैं आपको एक सीक्वेंस बताऊं कि कैसे प्रोटीन का प्रॉपर डाइजेशन होता है, so for example assume that this is a large protein molecule, something like this, ठीक है, assume करो कि ये क्या है? These are the large proteins which is broken down into the small peptides, ठीक है, peptides, या फिर इसको हम लोग क्या कहते हैं? पेप्टोन्स, या फिर क्या हो सकता है? प्रोटियोसेस. ओके, प्रोटियोसेस, these are the types of short पेप्टाइड चैन, ये बात आपको याद रखना है. ओके, so these are what? Short peptide chain formed after the breakdown or after the hydrolysis of the large protein molecule. Okay, so these proteases like intermediate stage, like if these peptones or the proteases are further broken down, it will form the individual amino acids. Ab samajh mein aaya? So what is the difference between protein, proteoses or the peptones and the amino acid?
Yes, sir.
Is it clear? सबको समझ में आया ये? Anyone? Yes? ऐसा कोई है जिसको ये डाउट है, मतलब still they are not able to understand this concept? ऐसा कोई है person? No? So now we will move to the next slide, that is digestion in the small intestine. So I think so we have completed some part of this, but again we know that there are 4 different types of cells are present, and different cells has a different function. Like the absorptive cells will absorb the nutrient from the lumen of the small intestine. Yes or no? ये अपने को पता है ना, goblet cell secretes mucus, and enteroendocrine cell secretes hormone, and this Paneth cell secretes lysosome. Okay, so these are the few functions of the layer of the cell. There is one more important point, is that there is one gland is like in this submucosa of the small intestine. One more gland is present, and that gland is called as Brunner's gland. Okay, remember this. There is one more gland present inside the submucosa of the intestine, small intestine. Okay, that gland is called as Brunner's gland. Okay. So I have given details about this in the later slide, but ये gland कहाँ present होता है? This is present in the submucosa of the small intestine. Okay. फिर नेक्स्ट यहाँ पे क्या है, ये भी आपको मैंने बताया था कि द डाइजेस्टिव जूसेस, देखो ये आपको मैंने बताया था कि इट कंटेन्स डिफरेंट काइंड्स ऑफ juices, like it contains the bile juice, it contains the pancreatic juice and the intestinal juice. Okay, ये सब मैंने आपको बताया था. Yes, you all remember this?
Yes.
याद है कि नहीं याद? So which factor, देखो इसमें important क्या है, this enterokinase, कहाँ गया this term? So this enterokinase will activate which enzyme? Trypsinogen. Okay, so once this becomes activated, this trypsin in turn activate all the other enzymes like the lipase, the amylase, the nucleases. So all these enzymes gets activated once the trypsin is formed. And how trypsin is formed? It is formed from the trypsinogen by the hormone or by this enzyme enterokinase. Okay, is it clear? पक्का समझ में आ रहा है? चलो फिर क्या है, बाइल. अगर आपको पता है कि बाइल जो है क्या, it is released into the duodenum by the opening which is called as a duodenal papillae. Okay, and that duodenal papillae contains opening of the ampulla of Vater, है ना? और वो जो opening को regulate कौन करता है? Sphincter of Oddi. Okay, so that all you should remember. तो ये क्या है? This bile contains bilirubin, biliverdin, bile salt, cholesterol. Okay, so these are the just chemicals which is present in the bile juice, which helps in the emulsification of the fat to form the micelles and activation of the lipase. And I have shown you this, na, ki how the micelle formation and how the absorption of fat globules takes place. Okay, so this image is showing the digestion as well as absorption of the fat globule. So again, I will just revise this. So this is a fat globule, okay, which is also called as lipids. So along with this bile salt, it is broken down or it is emulsified into small fat droplets, and then it is digested by the enzyme lipase, which will break down the fat, this small droplets into the fatty acid, that is a monoglycerides. Okay, and then this monoglycerides will enter inside a cell once. Okay, so it will enter inside a cell by the simple diffusion mechanism. Okay, because these lipids are what? These are the lipid soluble compounds. Okay, inside this cell, inside the epithelial cells or mucosal cells, special type of protein is synthesized which will cover the lipids, which is called as chylomicron. Okay, फिर chylomicron will leave the cell via exocytosis, and then it will enter inside the lacteals. Then this lacteals will transport the blood to the blood vessel. So this is how absorption and sorry, digestion and absorption takes place. So this part is showing the digestion, and this part is showing absorption of the lipids. समझ में आया सबको ये? Any doubt? ये क्लियर है? चलो ठीक है. So now this is the next concept, that is succus entericus. Succus entericus is what? The intestinal juice which is present in the duodenum or simply in the intestine. Okay, so this succus entericus, देखो आपको याद रखना है, the pH of the succus entericus is 7.8. Okay, 8. The pH of the succus entericus is how much? It is 7.8. So why it is 7.8? Because of the secretion of the mucus from the mucosa. और एक और यहाँ पे होता है जो म्यूकस along with the bicarbonates. देखो एग्जाम में कभी कभी ये पूछ लेता है कि this bicarbonate will make the medium alkaline. Okay, so this bicarbonates will make the medium alkaline. Okay, is it clear? And फिर ये देखो the different composition, like 99% is water and this remaining 0.5% is solid, and this solid is again categorized into organic compound and inorganic compound. ठीक है? And this organic compound is made up of enzymes and other organic substances. तो आपको याद कर रखना है the name of these enzymes which is present in the intestinal juice. तो एक enzyme कौन सा है यहाँ पर? Peptidase, aminopeptidase, okay, dipeptidase and tripeptidase. Okay, so tripeptidase क्या करेगा? It will break down the tripeptides. So for example, जैसे मान लो कि ये है, this is a tripeptidase ना, because it contains 3 different peptide bond. So they will break down this bond, tripeptidase. So ये enzyme क्या करेगा? This will break down the tripeptides. This will break down the dipeptides, and this will break down the peptides to form the amino acid. Okay. So these are the different types of proteolytic enzyme or the peptidase. और ये जो enterokinase है, this enzyme will activate, इसका function क्या होगा? बताओ, मैंने अभी बताया ना? ये enterokinase क्या करेगा?
Activate trypsinogen.
है ना? Activate trypsinogen to form the trypsin, and in turn that will activate other enzyme. ठीक है. फिर next यहाँ पे क्या है? The second amylolytic enzyme. मतलब these enzymes are responsible for the digestion and the digestion of the carbohydrate. ठीक है. तो carbohydrate की digestion में कौन help करता है? This amylolytic enzyme. Amylose मतलब क्या होता है? Like starch. ठीक है. So first enzyme is what? Sucrase. Sucrase क्या करेगा? It will digest sucrose into fructose and glucose. Maltose will digest this. This maltase will digest maltose. Lactase will digest lactose. Dextrinase will digest dextrine. Trehalase. So these all are the what? The different types of the carbohydrate present in the food. ठीक है. और एक एंजाइम कौन सा है? लिपोलाइटिक एंजाइम, और इसमें एक ही नाम आता है, दैट इज लाइपेज. ओके, LIPASE. सो दिस लाइपेज विल डाइजेस्ट लिपिड. ठीक है, इज इट क्लियर? यहाँ तक पूरा क्लियर है?
Yes.
एग्जांपल्स ऑफ यू एंजाइम्स देखो ना, बहुत ही इजी है. फिर एक और कुछ आता है ऑर्गेनिक फैक्टर में, जैसे कि म्यूकस हो गया, इंट्रिंसिक फैक्टर, डिफरेंसेंस. सो इंट्रिंसिक फैक्टर जो है किसमें हेल्प करता है? फॉर द एब्जॉर्प्शन ऑफ सम काइंड्स ऑफ विटामिन्स. Defenses किसमें help करेगा? Providing immunity, and mucus will make the medium alkaline, and it will also give protection to the mucus lining of the small intestine. है ना? So these are the functions of the other organic substances of the succus entericus. And the intestinal juice also contains inorganic compounds like sodium, calcium, potassium, bicarbonates, which makes the medium alkaline. Okay, chloride, phosphate, and sulfate. Okay, so these are the chemical composition of the succus entericus. And what is succus entericus? The intestinal fluid or the juice present in the intestine, okay, and the secretion of the brush border of the mucosa and the goblet cell. Okay, so the secretion of brush border of the mucosa and goblet cell is called as intestinal juice. इतना क्लियर है intestine के बारे में?
Yes.
Okay, then this is what हाँ ये तो क्या है? This is a Brunner's gland. Again ये present कहाँ पे होता है? This is present in the submucosa of the duodenum. Okay, and it secretes an alkaline fluid which contains mucin. Mucin is a protein which is present in the mucus. Okay, this is a type of protein which is present in a mucus. Okay, and it protects against the acidic chyme. What is acidic chyme? The chyme which is mixed with the gastric juice. है ना? So when the acidic chyme, like suppose this is the stomach, है ना? So this is the esophagus. So when the and suppose this is the duodenum. So when the food is coming from the stomach, so definitely the chyme will be acidic in the medium, है ना? So this chyme is acidic in medium. So to neutralize this Brunner's gland will secrete some mucus. Okay. So the secretion of this Brunner's gland helps in like protecting the mucosa of the duodenum from the acidic chyme. ठीक है? ये क्लियर है इतना? फिर नेक्स्ट यहाँ पे क्या है? Okay, this image I have taken from your textbook, simply from the NCERT, because they have given many example of the reactions. So even you can refer when you are referring this your textbook. So first, if you see in this reaction, I have told you na ki the proteins or the peptones or the proteases, because all the proteins are not broken down into the stomach. So the protein which is not modified, which is not broken down, so the proteins or the peptones or the proteases is digested by the different enzymes like the trypsin or the chymotrypsin or the carboxypeptidase. Okay, so these are the three different types of enzyme. Okay, this carboxypeptidase actually it removes carboxyl group from the peptide. Okay, so ultimately they will attack on this carboxypeptidase attacks on the carboxyl group of the peptides to make the dipeptides. Okay, so if possible, remember this name: trypsin, chymotrypsin, and this carboxypeptidase makes dipeptides. It makes dipeptides from the proteins, peptones, and proteases. Okay, is it clear?
Yes.
Next is the polysaccharide. Again, we know like there are different, there are two types of amylases there. One is pancreatic amylase. The second one is salivary amylase, but the basic function is same. The starch is converted into the disaccharides like the maltose. Okay, so this is the digestion of the carbohydrate into the small intestine. And for the lipids, again, we know that the fats are broken down into the diglycerides, then into monoglycerides. Okay, like they will remove the glycerol group by stepwise manner. So first they will, because fats are what? Fats are, they are the triglycerides, na? So first they will remove one glycerol chant from the site, then they will remove the second one, then the third one. So lipase acts on this. They will convert diglycerides and then the monoglycerides. Next is this, the digestion of the nucleic acid. Okay, any doubt in this particular slide?
No, sir.
Okay. In this, the nucleic acids, yes, simply we know that whenever we consume anything, then along with that, some nucleic acids also enters now. So for the digestion of nucleic acids, nucleases are present. So these nucleases will break down the nucleic acids into the nucleotides, okay, and then that nucleotide is further broken down into some enzyme into the nucleosides. And I hope I hope you know the difference between the nucleotides and the nucleosides. Pata hai kisi ko what is the difference between these two?
Yes, sir.
Yes, sir.
Just tell me in one point. Difference kya hai?
Nucleoside is sugar, deoxyribose sugar plus the nitrogen base.
No, that is not correct. Nucleoside is actually, it is a nitrogen base plus ribose sugar. It could be deoxy or the ribose sugar. So when the phosphate group attaches to the ribose attached to this, so what is nucleoside? It is nitrogen base plus the ribose sugar. So this is called as nucleoside. And when the phosphate group attached to the nucleoside, then it becomes nucleotide. Yes or no? Nitrogen base plus ribose sugar makes nucleoside, and when the phosphate group will attach, then it will form the nucleotide. Yes? You can check
Yes, sir.
these notes, then you will again understand. Okay, fine. So the next is what? Uske baad kya hai next? There are many enzymes that will digest the peptides into the amino acids. देखो जैसे यहाँ पे क्या है? The first one is dipeptides. तो उसके लिए कौन सा एंजाइम होगा? Dipeptidase that will form the amino acid. Maltose is broken down into the glucose plus glucose molecule. So here what is important? You should remember कि from one maltose we can get two glucose molecule. Okay? But from one lactose we can get one glucose plus one galactose. And from sucrose we can get one glucose plus fructose. So ये important है. This is one of the most important part. Because many times student gets confused कि मतलब what will form. So one maltose, and remember all these things like देखो ये maltose, lactose, sucrose, ये तीनों क्या है? These all are the disaccharides. ये सब क्या है? These are the disaccharides. Okay. So when they are broken down by the respective enzymes, so they will form the monosaccharides. So glucose, galactose, fructose, all these are what? They are the monosaccharides. But ultimately, all these disaccharides are, at least they are providing glucose. And what is different? The difference is that maltose will give two glucose molecule, lactose will give one galactose, and this sucrose will give one fructose. Itna clear hai? Yes?
Yes, sir.
Pakka? Chalo. फिर नेक्स्ट इज व्हाट? सम न्यूक्लियोटाइड्स. हाँ देखो, फिर जो न्यूक्लियोटाइड जो न्यूक्लिक एसिड से फॉर्म हुआ था, दैट इज कन्वर्टेड इनटू न्यूक्लियोसाइड्स. मैं इसके बात कर रहा हूँ. हालांकि देखो एग्जैक्टली आपके बुक में ऐसे ही मैंने कॉपी किया, सो एटलीस्ट दे शुड गिव दिस इंफॉर्मेशन हियर जस्ट बिलो दिस ना, बिकॉज we are learning so but they have given, it is okay. So now we are again talking about this nucleic acid digestion or the digestion of nucleic acid. ठीक है. तो यहाँ पे देखो जो न्यूक्लिक एसिड से क्या फॉर्म हुआ था? न्यूक्लियोटाइड्स. फिर यहाँ से क्या फॉर्म होता है? न्यूक्लियोसाइड्स. So first we will see how this nucleotide is getting converted into nucleosides. So which enzyme will convert? Nucleotidase, है ना? So this will convert nucleotides into the nucleosides. फिर this nucleosidase is further converted into sugar plus bases. दिख रहा है यहाँ पे? So it means that this nucleoside is made up of sugar plus bases. है कि नहीं? Even you can get clue from here. and nucleotide has additional molecule group, that is phosphate. ठीक है, तो जब फॉस्फेट ग्रुप वहां से निकलता है तो क्या फॉर्म होता है, न्यूक्लियोसाइड। सॉरी, तो उसके पास क्या होगा, फॉस्फेट ग्रुप भी प्रेजेंट होगा। ठीक है, so when the phosphate group is released तो क्या फॉर्म होगा, nucleoside, and when this is broken down it will form the sugars and bases.
So the phosphate group is removed from nucleotides.
Yes,
Okay, yeah.
so first they will remove phosphate group from the nucleotide to make a nucleoside. ये भी आपको याद रखना है कि what is the sequence of a breakdown of the nucleic acid. So first nucleic acid, okay, this is the first step, this is the second I can call it, and this is the third. Difference क्या है, so when the first they will break down the nucleic acid into a small unit that is called as nucleotide. Okay, then they will remove phosphate group from the nucleotide. They will remove phosphate group to form the nucleosides. Then they will break down into sugar plus nitrogen bases. So this is the digestion of nucleic acid. Clear है इतना?
Yes.
ठीक है, so this was about the digestion of nucleic acid. उसके बाद next is, हाँ ये भी important point था कि all the given digestion occurs in duodenum of our small intestine and simple substances are absorbed in the jejunum and ileum. मतलब ये सब जो आपने देखा ना digestion, देखो यहाँ पर एक और बाकी है, di and monoglycerides, they are also converted into fatty acids and the glycerol, and who will do this, the lipase. So all these reactions which we have discussed right now, okay, all these reactions occur in the duodenum of small intestine. Okay, this is also you have to remember, all these reactions occurs in the duodenum of small intestine. उसके बाद फिर क्या होता है कि as the food is passes, like again suppose this is the stomach and this is the duodenum. तो डाइजेशन कहाँ पे हो रहा है, यहाँ पर duodenum में. उसके बाद as the food is moving from the small intestine, the absorption of the simpler substances will take place. ठीक है, is it clear? समझ में आ रहा है सबको?
हाँ सर.
पक्का? चलो फिर we will
Yes sir.
again, क्योंकि this meeting is going to end. Okay, then we will continue the remaining part.